The only test right now that most labs are conducting to confirm the presence of SARS-CoV-2 virus is the Rt-Pcr test, which means the reverse transcription polymerase chain reaction test. So in today's video, we shall try to understand what PCR means, that is polymerase chain reaction, what is Rt-Pcr test, how is it conducted, and why it is called as a double-edged sword. But before that, let us briefly understand the COVID-19 disease. It is a respiratory disease caused by SARS-CoV-2 virus, which means severe acute respiratory syndrome coronavirus 2. More importantly, here SARS-CoV-2 virus is a RNA virus, which means it has only RNA as the genetic material. Like you all know, on 30th January 2020, World Health Organization declared this outbreak as a public health emergency of international concern and a pandemic on 11th March 2020. Like I mentioned earlier, COVID-19 is a respiratory disease. Hence, to confirm the presence of SARS-CoV-2 virus, specimens are collected from respiratory tracts. And once these specimens are collected, they are sent to labs to confirm the presence of SARS-CoV-2 virus using the Rt-Pcr test. Now, let us try to avoid the technical and scientific details of the Rt-Pcr test. Instead, we shall try to understand it from a common man's perspective. Firstly, concentrate on these three terms, that is polymerase chain reaction. Every cell has a genetic information. It is a set of instructions on how a cell should live, what functions should it perform, and eventually how it should die. Basically, this genetic material is found in two forms. One is ribose nucleic acids, that is RNA, and the second one is deoxyribose nucleic acid, that is DNA. DNA here is a two-stranded molecule that is found in almost all organisms, that is animals, plants, and most of the viruses, while RNA is a single-stranded molecule and differs in functions from that of DNA. Here, PCR or the polymerase chain reaction is a method widely used in molecular biology to rapidly make millions or even billions of copies of DNA samples. Basically, a PCR kit works like a Xerox machine, making multiple copies of a base DNA in no matter of time. So scientists here take a small amount of DNA and amplify it multiple times to study in detail. So PCR here cannot be directly applied or conducted to confirm the presence of SARS-CoV-2 virus. Why? Because this virus is a single-stranded RNA virus. Thus, this poses a problem in the traditional PCR technique. Here, only DNA can be detected and amplified. Therefore, we need to convert the RNA of the coronavirus to DNA. We call this as reverse transcription. Similarly, when a DNA is converted to RNA, it is called as transcription. So, we call this as reverse transcription. Hence, using the technique of reverse transcription with the polymerase chain reaction method, we call it as reverse transcription polymerase chain reaction test. But how it works? Firstly, the sample is collected from the parts of the body where the coronavirus gathers, such as the person's throat or nose. Further, this sample is treated with many chemical solutions to extract only the RNA sample, leaving behind proteins and fats in those samples. But wait, this extracted RNA is a mixture of a person's own genetic material and, if present, the genetic material of SARS-CoV-2 virus. But first, the extracted RNA is reverse transcribed to DNA using a specific enzyme. And from here, the extracted and converted genetic material is amplified and copies or similar copies are made in millions and in billions. A real-time Rt-Pcr setup usually goes through 35 cycles, which means that by the end of the process, around 35 billion new copies of the sections of viral DNA are created. During the process, specific short fragments of DNA are added to these samples. The basic job of these fragments is to attach themselves to specific sections of the viral DNA if present. Additionally, and more importantly, these fragment DNAs also contain marker labels, which are then used to confirm the presence of SARS-CoV-2 virus. And further, as new copies of the viral DNA sections are built, the marker labels attach to the DNA strands and then release a fluorescent dye. This is then measured by the machine's computer and presented in real time on the screen. When the amount goes over a certain level of fluorescence, this confirms that the virus is present. This Rt-Pcr test is said to be a highly sensitive and highly specific test to confirm the presence of SARS-CoV-2 virus. It usually takes 3 to 4 hours to complete the whole process. And compared to other available methods, real-time PCR technique is faster and also has lower potential for contamination or errors. Last week in Hindu's editorial section, an article was given which compared the Rt-Pcr technique to a double-edged sword. It is because, as per this article, both the boon as well as bane of PCR testing are in its capacity to amplify even one viral gene segment in the sample to generate a detectable signal, that is a positive test. And it is a boon because it accurately detects the presence of virus, but a bane because it is prone to false negative and false positive results. The article says when a laboratory handles several samples, there is a chance of cross-contamination. Essentially, the article argues that if a gene segment from confirmed sample falls into another tube sample, then it might show false result. And to avoid such instances, a laboratory technician's proficiency is integral to quality assurance. And for more reliability, only laboratories under quality assurance should do testing. And even when a false positive result is suspected, the doctor should immediately alert the authorities, who in turn should get the subject retested in an accredited laboratory. In case of discrepancy, the laboratory concerned must be closed and checked for compliance with protocols and record keeping.